même temps que nous. On faisait 2 travaux parallèles et on travaillait avec une laboratories qui sont le premier grand laboratoire d'Acadie où travailler avec là-dessus. Qui sont un endroit merveilleux et ils ont une contribution très très importante dans la connaissance de Coxiella, de la fièvre Q dans ce laboratoire. Je suis très ravi qu'ils viennent nous faire un séminaire. All good.
Doctor will all say good things about me. I'll give it a second. Well, it's truly a great honor to be here in Marseille among so many leaders in the field of rickettsial research, doing this fabulous work here over the last 30 or so years. This is where I come from, the Rocky Mountain Labs in Hamilton, Montana, USA. And I'm just showing a picture of the campus. This is actually some new construction that we've had recently that involves the construction of a biosafety level 4 high containment lab for viruses such as Ebola and other hemorrhagic fever viruses. This is our level 3 facility. This is the older part of campus. I actually have a lab in this building here. So today what I'd like to tell you about are some of the advances we've made in studying this difficult form of the obligated intracellular bacterium, Coxiella burnetii, and focus on some of the new genetic techniques we've developed that are helping us describe some of the virulence factors of this pathogen. Since you're all very well versed in the field of rickettsial diseases, I won't spend a lot of time on background, but let me give you just a few slides. This is actually the first case of acute Q fever ever described in the United States, and in the literature, this patient is referred to as patient X. You can see that they have a high spiking fever that lasts for about 2 weeks. This person had other flu-like symptoms, and what makes patient X really interesting is who it was. It happened to be this distinguishing looking fellow who was none other than, at the time, the chief of the Division of Infectious Diseases at the National Institutes of Health. Later went on to become the director of the National Institutes of Health, a guy named by the name of Rolla Dyer. He actually wrote up his own case report entitled Dyer et al. in the Public Health Report. So how did Rolla get Q fever? It was actually through a visit to the Rocky Mountain Labs. Here's the lab shown in the 1940s. And he became interested during this visit in a new pathogen that had been described at the lab that was referred to simply as a 9-mile agent. It had been isolated from a tick as part of a epidemiological survey on Rocky Mountain spotted fever. But the pathogen in a guinea pig did not cause a disease that resembled spotted fever. It caused something really quite different. But to make a long story short, Dyer became infected with a 9-mile agent. But he was also a rickettsiologist, and he recognized, and he was familiar with a new clinical entity that had been recently described in Australia that was being referred to as Q fever. So through the exchange of reagents with the investigators at the time in Australia, mainly McFarland Burnett, it was defined that the Q fever agent in Australia and the 9-mile agent in the United States were actually the same pathogen, and they both cause a disease called Q fever. And here are some of Burnett's early drawings of the intracellular nature of C. burnetii and mouse melanocytes. A fact that many people don't know or recognize is that McFarland Burnett actually went on to win a Nobel Prize in the early 1960s for his work on immune tolerance. So a very accomplished scientist. So Rolla had a classic case of acute Q fever. It generally manifests as a severe flu-like illness. One reason he became so easily infected is because this is a highly infectious organism with an infected dose protein 1. It's transmitted by aerosols. It has spore-like environmental stability. When we combine all these characteristics together, you have what at least the U.S. Centers for Disease Control and Prevention considers a category B biothreat with the potential for illegitimate use by a And that is the very prevalent animal reservoirs of Cibernetti, shown here. In terms of human infection, it's primarily our association with domestic livestock. There are literally millions of dairy cattle infected with Cibernetti in the United States. Goats and sheep are major animal reservoirs. The cat pops up in urban outbreaks, acute fever every now and then. But nonetheless, the animal reservoirs are really quite extensive. So acute fever is a classic zoonosis, and that has been evidenced clearly during this outbreak in the Netherlands that some of you may be aware about. It's considered the largest acute fever outbreak ever recorded. Over 4,000 cases to date. It's now subsiding due to control measures. And one of those control measures is actually to cull or kill over 40,000 pregnant dairy goats because they had such high levels of infection. Okay, on to some cell biology. During natural infection, Cibernetti has a tropism for cells of the mononuclear phagocyte system, such as macrophages. It resides in an acidic parasitophorous vacuole and requires the acidity of this compartment to activate its metabolism. If you take purified Cibernetti and put them in a test tube at neutral pH, with a lot of nutrients, they're essentially metabolically inert. They require a pH of around 5 to become metabolically active. And they found a niche within the host cell that provides that acidity. It's historically been considered an obligate intracellular bacterium, and the reason for my use of the word historically will become evident. I think really now we need to consider this a facultative intracellular bacterium. But a consequence of this obligatism over time is that it's been very difficult to develop genetic methods for any obligate intracellular bacterium. When you have a host cell as a growth medium, it's difficult to clone, it's difficult to select for transformants, and so on. As a consequence of poor genetics, really to date, the LPS is still the only genetically defined virulence determinant of Cibernetti. Virulent organisms make a full-length LPS with a complete O antigen side chain. Avirulent organisms make a very truncated LPS. So LPS is critical for virulence. So here's an example of some infected macrophages, and I always enjoy showing this slide because I'm always impressed with the immense parasitic burden that these cells can withstand. So here's a J774 mouse macrophage stained with a metastain, red, and perhaps 1,000 microorganisms in this vacuole. It's essentially grown to encompass nearly the entire host cell cytoplasm, a little bit of cytoplasm left, the nucleus pushes the periphery of the cell. Here's a time-lapse video loop of, again, these are primary human PBMC-derived macrophages, and you can see perhaps thousands of Cibernetti in this vacuole. This is the cytoplasm that's left, and then the nucleus is this phase-bright material pushed to the edge. So these are essentially bags of Coxiella. So there's obviously some major subversion of host cell processes that enable the development of this replicative niche. And we're very much interested in knowing how that process works. But again, as I alluded to, an obligate intracellular nature imposes some very significant experimental constraints. And so we decided about 5 years ago to try and solve this problem and possibly figure out a means to grow this organism outside of its host cell. That is to recapitulate this environment to a close enough degree that the organism would actually replicate in a test tube. And we thought we might have a reasonably good chance based on the genome that was published in 2003, and it showed that Cibernetti is fairly metabolically sophisticated for an obligate intracellular bacterium, as a genome that is roughly twice the size of most obligates. There's little genome reduction, most of the pathways, the central carbon metabolism and bioenergetics are attacked. So you can't look at the genome and go, here, look at all these metabolic lesions, that's why it's an obligate. We just didn't see that. So to make a huge amount of work, turn this into a very short story, a very talented postdoc in the lab, Anders Omsland, developed a buffer called a medium called acidified citrate cysteine medium that supported prolonged Coxiella metabolic fitness. And what I mean by fitness is the ability to take Coxiella purified from host cells, put them in this buffer, and are they able to sustain protein synthesis and to make ATP? And this buffer was really quite good. And in a nutshell, what it is citrate. And citrate is found to be quite good at maintaining fitness. And of course, we need a low pH of about 4.5. It had relatively high sodium and chloride levels, higher than you would find in the cytoplasm of the host cell. 2 rich amino acid and peptide sources. The organism appears to prefer amino acids for carbon and energy. And then a high level of L-cysteine, and this is a trick we stole from the Legionella field. It's been known for a long time that to grow Legionella, you need to add a lot of L-cysteine to the media, and it's not really clear what they need that cysteine for. So this was all good, and we did have a buffer that was quite good at activating the organism. But when we looked at an increase in genomes that would indicate replication, we never saw any increase. So they weren't replicating in this media. And Anders had been obsessed with this bit of bioinformatic data that showed that Cibernetti has the ability to synthesize a branched respiratory chain. That is one that either encodes cytochrome O, which is used under highly aerobic conditions by gram-negatives, or a chain that can switch to cytochrome BD, which is used by gram-negatives under microaerobic conditions. So it looked like Coxiella could actually respond to oxygen tension. And the way Anders tested that was to use something called a biolog phenotype microarray. And what this is, it's sold by a company, and you're 96 well plates with a different substrate in every well. And these substrates are primarily amino acids and TCA cycle intermediates. And what you do is you inoculate the well with a buffer containing your organism of choice, a minimal buffer. And if the substrate is oxidized, the tetrazolium dye in the well is reduced and turns purple. It's black and white version here. But nonetheless, when Anders tested Coxiella using these plates at 20%, 5%, and 2.5% oxygen, the lower the oxygen level was, the more substrates were oxidized. So it appeared, at least via this assay, that Coxiella really preferred a low oxygen environment. So we put ACCM media together with low oxygen, and it came up with this really remarkable result in that we got very robust growth in the organism in only a 2.5% oxygen environment. If you try to grow these organisms at ambient oxygen, 20%, what we're breathing now, they just don't replicate. So this is 3 logs of growth over 6 days with a log phase generation time of about 9 hours, which is a little bit faster than the generation time that we see in a very permissive cell type like a Burrow cell. And Anders has since gone on to further modify this media to generate something called ACCM2, where it's a simple change, where instead of fetal bovine serum, we add a compound called methyl beta cyclodextrin. And when we do that, we increase the yield. We get about 4 logs of growth in about 4 days and generation time of about 5 hours. So now we're looking at generation times about twice as fast as what we get in the host cell. So this is our medium of choice right now, and it also results in a more stable viability of Cibernetti during prolonged incubations. Okay, so what is azenic culture allowing us to do? Well, for the first time, it's allowed us to actually characterize what we refer to as the infection-specific transcriptome. And so what we've done here is do a microarray with organisms cultured at an ACCM for 4 days or macrophages for 4 days. So they're about the same growth stage. And then just giving you a snapshot of what genes are upregulated in macrophages, this is what we observe. The organisms appear to be under a fair amount of oxidative stress because we see a number of oxidative stress proteins upregulated, or genes upregulated in macrophages, such as alkyl hydroperoxide reductase is up 25-fold during growth in macrophages. We also see a number of transporters and permeases upregulated, and that to us suggests that intracellular growth is within a somewhat of a nutrient-limited environment. And that makes sense when you think back to those generation times, that we can get much faster generation times in a complex media. A number of lipoproteins are up, some virulence proteins have been described, and Coxiella is close, relative Legionella are up 10 to 20-fold. Then, of course, we have all the hypotheticals, and actually the most upregulated protein is CDU1932, which is very basic and has some histone-like properties, and that's up 38-fold in macrophages. So we're starting to sort through this list and picking some interesting candidates to look at more closely. It's also allowed us to identify what I refer to as the ACCM secretome. What proteins are actually secreted by Coxiella into this medium. And we can adjust the medium to allow us to do mass spec. And this is just a partial list of about 30 proteins that we've identified are secreted by the organism, presumably by a single peptidase type 2 secretion-like system. And again, we can see some oxidative stress proteins. Mostly these are hypothetical. So we're left with trying to figure out what this collection of hypothetical proteins is doing, because presumably these are also secreted into the vacuole lumen and are modifying something in that vacuole lumen that allows cibernetics to proliferate intracellularly. And finally, what azenic growth has really accelerated is our ability to develop genetics for Coxiella. So here's a paper we published about 3 years ago where we were able to transposon mutagenize Coxiella using the HIMAR1 transposon. And here's a particular mutant expressing M-cherry in viral cells. So this is a host cell-based transformation protocol. We didn't have our media at the time. In a very tedious, lengthy procedure, it takes 2 to 3 months to actually derive a clone, a single clone, from an experiment like this. So using host cells for transformation experiments is just very, it's very unruly. It takes quite a long time to test new technologies and so on. But now with our azenic growth methods, we figured out how to get Coxiella to actually form colonies on ACCM agarose plates. It has to be agarose, it can't be agar. And the colonies are really small. On a good day, they might hit 0.5 millimeter. Sometimes they're smaller, but you can pick them and you can expand them and you can show that they're clonable. So this is an excellent way to clone. And now we're looking at 2 to 3 weeks after a transformation experiment to actually derive a clone. And it also allows us to recover non-infectious mutants. Because if you have host cells as your growth medium, anything that lowers fitness for growth in host cells, you're probably not going to recover. Okay, I'm just going to go walk through some of these tools real quickly. What we've been able to do is define RSF1010 ORI-based shuttle vectors that work quite nicely in Coxiella that are compatible with the endogenous plasmid. We've established a method for inducible gene expression that uses anhydrotetracycline. It's very nice to turn genes on and off in Coxiella. And here's an example where we have M-cherry protein under control of the TET promoter. Cells infected for 3 days. We add tetracycline. You can see this vacuole becomes red after about 8 hours. The organism's make M-cherry after induction. And we can see this by immunoblot as well. So it's very tightly regulated. It works quite well. We've figured out a way to use a TN7 transposon for single copy site-specific in-cis complementation experiments. And what's nice about this transposon is that it inserts at a single site in the chromosome at an intergenic region right downstream from a housekeeping gene called CLIMS. So the insertion has no effect on cybernetic growth, and it gives you a single copy of your gene of interest at a known site in the chromosome. So it uses extensively for complementation experiments. So the remaining piece of the puzzle, obviously, is the ability to do targeted gene inactivation. Here's a gene you're interested in. I want to make a mutant, and I want to phenotype that mutant. And we've since developed a couple of methods to do this, and I'll show you just one very quickly. And it's really old school E. coli Pseudomonas gene knockout technology. It's called a loop-in, loop-out procedure where you use the gene SACB as a counter-selectable marker. And SACB is from Bacillus subtilis, and it encodes an enzyme that polymerizes sucrose into toxic fructose molecules. So if you grow an organism that has SACB in the presence of sucrose, that's a toxic environment for the organism, and it needs to get rid of that gene. So it forces a recombination event that you're after. So here's an example of how we do this. We're going to knock out a gene called .A. Replace .A with a kanamycin cassette, resistance cassette. We clone the 5' and 3' flanking regions of .A. We transform and select for a single recombination event. In this case, it's recombining between the 5' regions. Then we resolve the co-integrant by growing this transformant in the presence of 1% sucrose. And this was actually a little tricky to develop because 1% is much lower than the percentage of sucrose used for E. coli or other pathogens or other bacteria. Typically it's 5%, 10%, oftentimes it's 15%. But Coxiella, expressing SACB, is exquisitely sensitive to sucrose. So it turns out 1% works quite nicely. And what we're driving here is a recombination between the 3' regions. We also have kanamycin in the media, which will select for a can mark deletion. These guys recombine, resolve the co-integrant, and this is what we end up with. A kanamycin cassette in place of .A, and we can show that's happened by southern blot. So just to give you a little bit of information on what .A is, .A is actually a component of the organism's type 4 secretion system. And it's a .ICM type 4 secretion system, very similar to one that is produced by Legionella pneumophila. .A is thought to reside in the inner membrane, and these secretion systems are designed to translocate proteins with effector functions directly from the bacterium into the host cell cytoplasm, where they can modulate a variety of different functions. And we know that Coxiella is actively involved in host cell manipulation in a way that promotes biogenesis of its replication vacuole. And here's a simple experiment, but I think it really illustrates nicely what I'm talking about. If you infect, in this case, it's viral cells with Coxiella in the presence of chloramphenicol, which inhibits Coxiella protein synthesis. And these cells have been infected for 48 hours, and you stain lysosomes, essentially, or acidic vesicles, with acridine orange, so those appear orange, and you can counterstain the cells with a DNA stain, which stains the nuclei green, and it's also going to stain Coxiella as well. What you see in the chloramphenicol treated situation is you have individual organisms in tight-fitting acidic vesicles that are scattered throughout the cytoplasm. And Coxiella actually looks a bit yellow here because it's the merge of the green and orange colors. So this is what you observe when you treat with chloramphenicol. This is the wild type situation. You typically have one large and rather spacious vacuole. And what it tells us is that Coxiella needs to make protein to promote homotypic fusion between these vesicles to generate this one large vacuole. So making proteins that are directing the cyclic fusion. You can actually take the chloramphenicol off after a couple of days, and these guys, these bacteria are not dead. They'll simply start to make protein, and these vesicles will fuse, and you'll end up with something that looks like this. So our DADA mutant is completely deficient in the ability to infect mammalian cells. And that correlates with the inability to secrete known type 4 effector proteins. So these are a couple of proteins that were identified earlier by us. We plugged them into this DADA mutant, and you can see that we're using a translocation with an assay I won't go into, which involves the translocation of kelmodulin activated adenylate cyclase. But nonetheless, if you look at the cyclic AMP levels of wild type C-renity versus the mutant levels approach that of the negative control. It's completely unable to secrete these effector proteins. And that correlates with the inability to grow intracellularly in a very permissive cell type. So here we have a typical 6-day growth yield in viral cells of wild type of about 1,000-fold, the complemented mutant of about 1,000-fold, and then the mutant, which you usually see is about 3 or 4-fold increase in genome equivalence. We think what happens is the mutant is able to undergo a round or two of division, and that's it. And in ascenic media, there is no growth defect. It grows just fine. So it doesn't need the secretion system to grow in host cell-free media. And we can observe this null growth phenotype by immunofluorescence, where we have a wild-type organism showing this one large red parasitophorous vacuole. We have the mutant, again, this individual organisms in these tight-fitting lamp-positive vacuoles that don't fuse, and then the complemented mutant. Okay, so we've now generated roughly knockouts in about 20 genes. And we're starting to do some serious phenotyping on what the phenotypes of these knockout strains are. And I'm going to go into some detail on this particular knockout, because I think it's very preliminary, but it's very interesting. I'd like to know what everybody thinks about our ideas on this particular knockout. It involves cholesterol in a gene called CBU1206. So we published a paper in 2006 that demonstrated that the Coxiella vacuole membrane appears to be very enriched for cholesterol. And most host cell cholesterol is found in the plasma membrane, but when you look at this parasitophorous vacuole, shown here, stained with a fluorescent cholesterol binding compound called philipin, you see very strong fluorescence. And here's Coxiella that are expressing young cherry protein, and again the merge. So based on this assay, the vacuole membrane appears to be cholesterol rich, but the caveat here is that philipin binds all sterols. So it can bind cholesterol sterol intermediates, but the presumption has always been that this is cholesterol. Then our group and also DDA's group published papers that showed that if you treat infected cells with inhibitors of cholesterol biosynthesis or uptake, you can have a pretty dramatic negative effect on replication and vacuole generation. And here's an example of a panel of inhibitors that inhibit either uptake or biosynthesis. And for example, lovastatin, one of the statin drugs, you get vacuoles roughly half the size of wild type and about half the replication. And we have some of these drugs that you get absolutely no replication. But a caveat here is that many of these drugs have off-target effects. And also, they have side effects that you really don't want. So, for example, U1AH6 inhibits both biosynthesis and uptake, and it also is known to intercalate into membranes and disrupt the lipid architecture in that area. So these drugs are problematic too in terms of defining a role for cholesterol in host pathogen relationships. So we decided a year or two ago to look at this a different way. And the model we used was mouse embryo fibroblasts from a knockout mouse that is deficient in an enzyme called DHC24, which encodes a delta 24 reductase, which is considered the final reduction step in cholesterol biosynthesis. So these mice, it took us a couple years to get these mice, and these mice, instead of cholesterol in their cellular membranes, have this precursor called desmosterol. And it's really, the only difference is the reduction. This bond is not reduced to make cholesterol. So what we needed to do was actually establish a real cholesterol-free cell line. And the way to do that is to make the heterozygote and then genotype the fibroblasts that we get from embryos as to whether or not they're actually DHC24 plus or DHC24 minus. Then we can adapt these fibroblasts to growth in serum-free basal media. So in this case, the homozygous wild type will still have cholesterol because they can make it biosynthetically. But the knockout, since there's no exogenous source of cholesterol, is truly cholesterol negative. And then what we can do with the knockout cells is we can add back known amounts of cholesterol and do basic titration experiments in that way. So here's some HPLC that shows that indeed our adapted mouse embryo fibroblasts are truly negative for cholesterol. Here's a wild type with a cholesterol peak. Here's our mutant adapted to basal media with desmosterol. Here's the mutant with some cholesterol added back. You get pretty much a 50-50 mix of cholesterol and desmosterol. We also wanted to know whether or not these cholesterol minus MEFs behave like a cell that lacks cholesterol. And it turns out that desmosterol cannot participate in the formation of lipid rafts. And if you know what lipid rafts are, they are membrane microdomains that are rich for cholesterol and single lipids that concentrate signaling molecules and attenuate the activity of those signaling molecules. So they thought to be very important to cellular signaling. And we looked at lipid raft function using this assay, where we're assaying the uptake of cholera-toxin by a GM1 granulocyte that's known to reside in lipid rafts. So here we have MEFs, where you've added back the cholesterol minus MEFs, where we've added back cholesterol. And this is an aminoperoxidase assay, where basically a cholera-toxin positive endosome is basically essentially a black dot. And when we score these MEFs, we can show that 80% of those MEFs that contain cholesterol have greater than 10 cholera-toxin positive endosomes. And that's really different from what we see with the truly cholesterol minus MEFs, where 9% have less than 10 cholera-toxin positive endosomes. So they appear to have very deficient lipid raft function. So now what happens when we infect these cells? Well, what we find is when we infect with Coxiella, the actual uptake assay, they appear to enter much more efficiently with cells containing cholesterol. So in the cholesterol minus MEFs, we see about a 30 to 40% decrease. And that suggests that Coxiella is entering cells via a lipid raft mediated process, and it's actually in agreement with some of the Jean-Louis data showing that they engage the integrin receptors, which are known to be lipid raft receptors. But what's interesting is once they're internalized, there's no defect. There's no defect in the growth yield or the generation of the vacuoles. So here are peristrophus vacuoles in the presence of cholesterol or the absence of cholesterol. There just really isn't any difference. And if you look at the actual kinetics of growth, they appear to be similar as well. You get fewer in, but the ones that get in grow just fine. So that was a surprising result. So one thing we thought about is that Coxiella actually encodes a homolog of eukaryotic delta 24 reductase. This is one of these interesting genes that DDA is interested in, that probably Coxiella acquired from in some association with amoeba. And it turns out that they have this delta 24 stellar reductase that has the highest identity with a homolog from Mygleria gruberi, which is a soil amoeba, and has very good homology with other eukaryotic reductases, such as that from Saccharomyces cerevisiae. So we wanted to know, is this enzyme actually a functional delta 24 reductase? And we used a yeast system to look at this because it's easy. And yeast actually would encode a protein called ERG4, which is their equivalent of the delta 24 reductase. And these ERG4 mutants are highly sensitive to go in different compounds, one being Grafeldin A, which disrupts the Golgi apparatus. So here you can see wild type, no problem, they can go in the presence of Grafeldin. The ERG mutant has a strong growth defect, complemented back with ERG4, you rescue the mutant. The mutant is not rescued with the human delta 24 reductase, but it is rescued with the Coxiella delta 24 reductase. So thus it appears from this assay that this Coxiella delta 24 reductase is actually active and can act on yeast precursors. So I should mention that yeast don't actually make cholesterol. They make something called ergosterol, which is chemically very similar to cholesterol, but a little bit different. But it turns out that we can prove that the Coxiella reductase is actually converting the ergosterol precursor to ergosterol by doing HPLC. So here's ERG4 mutant, not complemented, ergosterol precursor. We complement back with the ERG4, we get ergosterol back, no complementation with a human reductase. And then the ERG4 from Coxiella burnetii, it's not a lot of ergosterol, but it's definitely there, and it definitely rescues the phenotype. So Coxiella's delta 24 reductase is active. So the obvious question was, maybe the ability of Coxiella to grow in these cholesterol minus MEFs is that this delta 24 reductase is actually converting the desmosterol to cholesterol. It's compensating for the defect in that cell line. But when we look by HPLC, we don't see that. So here's again some the wild type MEFs making cholesterol, the knockout MEFs making only desmosterol, the knockout MEFs in the absence of cholesterol infected with Coxiella, we don't see any cholesterol. So now we're left with what is the function of this enzyme? Of what benefit can this be to Coxiella? And that brings us back to the genetics. We were able to knock this gene out using that loop in, loop out procedure I told you about. Here's the PCR showing that the gene truly is deleted, and that allowed us to look at the phenotype of the mutant. So the first thing we do with these mutants is to ask whether or not they grow normally in host cell-free media. They grow the same as wild type. There's no defect. We get our roughly 3.5 log fold increase over 4 to 6 days. But the knockout does have a fairly strong growth defect inside mammalian cells. So this happens to be HeLa cells, and if you compare it to wild type, you get these very small vacuoles at 3 days post-infection. And if you actually look at the size, you measure the size of the vacuoles, they're less than a quarter of the size. So it has a very strong growth defect. And these are cells grown at 10% fetal bovine serum, which would be about 30 micrograms per ml cholesterol. And we see the same characteristics in macrophages. So these are J774 macrophages. Very small vacuoles. If you look at the genome numbers, we're getting about 10% of the genome numbers with the mutant compared to the wild type. So then we went back to our MEFs, our cholesterol-free system that we can add back cholesterol. And we looked at the mutant in the absence of cholesterol, the vacuoles appear normal. When we add cholesterol back, either by, in this case, by adding 10% fetal bovine serum, we get back that small vacuole phenotype. And we can do this in a more controllable fashion by just adding known amounts of cholesterol back to the cholesterol-free MEFs, which is what's done here. And if you look at the MEFs with no cholesterol, the wild type and the knockout essentially grow the same. If you add cholesterol back, again we see a strongly significant difference between the vacuole size of the mutant versus the wild type, and we don't see that dramatic difference if we add back bismosphorol. So our conclusion from this is that the mutant appears to be hypersensitive to the presence of host cell cholesterol. And again, we can show that this is, again, looking at genome increases, and what we've done is normalize the genome numbers of the mutant grown in the presence of no cholesterol, and then grown with 2.5% micrograms per mil of cholesterol, 5 micrograms per mil of cholesterol, or 5 micrograms of bismosphorol. We see dramatic effects when we add cholesterol back, a little bit of effect when we add bismosphorol back, but it's not statistically significant. And again, this effect is only associated with growth in host cells. So if we add 5 microgram per mil back to ACCM media, or bismosphorol, or have no cholesterol, which is this, they all grow identically. So we have some ideas about this. I'm not going to be able to end the story today because I don't know the end. I don't know the end of the story. So what is the function of Coxiella's delta 24 cell reductase, and why does a knockout display growth defects in cells containing cholesterol? So one hypothesis is that the 1206 enzyme is modifying cholesterol in some fashion that benefits the pathway. And it could be something as simple as a reverse reaction, oxidizing cholesterol back to bismosphorol. And this, in some fashion, perhaps alters the membrane fluidity in some way that facilitates type 4 secretion. That's our working hypothesis. And we're taking this from the Helicobacter literature. And it's known that Helicobacter produces an enzyme called CAPJ, which actually is a cholesterol alpha glucosyl transferase. It puts glucose on cholesterol, and it really changes the behavior of that molecule in the lipid bilayer. It increases the fluidity, and is thought to augment the membrane attachment and assembly of the functional type 4 secretion system by that pathogen. So what we think might be happening is that 1206 is important at regions of Coxiella parasitophorus vacuole membrane. And it's there to modify this membrane in some fashion to facilitate the construction of these type 4 secretion needles and the subsequent secretion of effectors into the cytoplasm. And without this enzyme, this process is deficient, and you get this strong negative growth phenotype. So that's where we're heading with that, and I'd love to hear what else people might think about that idea, but we think it's a good one. So I'm going to summarize what I told you today, and first we can substitute fetal bovine serum with methyl beta-cyclodextrin and ACCM and get dramatic enhancement of azenic growth. We think what this molecule is doing is probably sequestering toxic fatty acids that might be released by the organism. Transcriptome analysis reveals upregulation of oxidative stress genes during intracellular growth. So I actually think the Achilles heel of Coxiella, the way Coxiella dies, is by oxidants. If you want to kill Coxiella, hit them with superoxide and nitric oxide, and they just don't know how to deal with it. Azenic growth allowed identification of a complex non-type 4 Coxiella secretome predicted to modify the PB lumen. Our host cell growth has really rapidly accelerated our ability to do basically everything we need to do now genetically with Coxiella. We pretty much closed the loop on our genetic tools. We're not really seeking to develop any new ones. We have what we need. The Dottie mutant is incapable of intracellular growth. This is predicted to be absolutely virulent in an animal model. Wild-type Coxiella grows normally in the absence of cholesterol. However, this delta 24 steroid reductase appears to be hypersensitive to host cell cholesterol. And we think that has something to do with modifying local regions of the parastrophes factor membrane. All right, so I'm going to, how do I do this? I'm going to end and introduce some of my lab members. Paul Bear has developed all the genetics and growth and molecular biology. Anders from Norway, great addition lab, worked very hard to develop Azenic media. Dale, a technician, has been with me for years, and Diane, another technician. Dan Hoff is now at the University of Arkansas, working on type 4 secretion. I didn't talk about Jeff's work today. He's worked on dendritic cells. And Stacy has done all the cholesterol work. So I will end there and take any questions.
There is one of your work that you have done that we have also done, but after this time. That wasn't the use of statin on the size of the vacuum. Do you think that this has something to do with the use of cholesterol? Let's say
Do we think it has something to do with the use
of cholesterol? Because the statin, when you incubate the Coxiella because the cell is changed in an RNA, it's changed the size of the vacuum.
Right.
Do you think that this has something to do with also?
Well, it's either, I mean, the problem with the statins is they inhibit early in the pathway. So it's not clear, there's a number of sterols that are made until you get to cholesterol. So the assumption is that the growth defect is because they don't have enough cholesterol in their factor membrane. I'm not so sure that's the whole story. It may be actually one of the precursors that is necessary for.
What we've seen is mainly that the size, they could not increase the size of the vacuole as much. That's what's the main thing that we found.
Yeah.
I know, do you find the same thing? Yeah.
Well, yeah, the vacuoles definitely don't, but it does also correlates to poor replication.
Did you check that this is something that we were looking at? Did you compare the bugs growing clinically to that grows within the cells? Morphologically?
By EM, yeah, we've looked at them. They don't look they look the same. So, I mean, at that resolution, we can't really tell the difference. We've done EM on the colonies as well. And what we see there is it looks like they've developed into that small cell variant form. And that's maybe one reason I'd like to, maybe you can figure out a way to get those colonies bigger. Because they're so small, they're like a grain of salt, and they're really hard to work with. And I don't know what limits the growth. Why do they stop? So,
well, in general, nobody knows why colonies stop to grow.
Yeah, these are very.
There is a lot of.
Now, I have some ideas that maybe the local microenvironment that is neutralized. That would be one idea.
Yeah. Some
or
people
this
say
is a thing.
Lorian was saying, Lorian, I don't know if you remember Lorian. Lorian was a great guy. And he worked for years on many of the staff to try to understand why there is a definite size of a colony. His main opinion was based on physical contention. He calculates the weight that one bug has to support with the size of the colony, and he considers that there is a maximal weight that you can support, and when it's come, you cannot grow anymore. So I think it was mainly a physical condition.
One thing I didn't mention is that those colonies actually grow within the agarose. It's an overlay. So we can't get them to grow on the surface. And I think that, again, is indicative of the need of Coxiella being an acid bath. You need to have acid around them. You can't just streak them on top and get it to grow. And the fact that they're in an overlay may also inhibit the actual size of the colonies.
Okay.
Thank you.